Hi everyone, we are going to get started. So my name is Steph Bellman. I'm one of the trainees in the Infectious Disease Across Scales training program, and today I'm extremely excited to introduce Maria Diuk from Columbia University. Dr. Diuk obtained her PhD from UCLA in biology, studying anophelines and malaria transmission in Mali. She then completed her postdoc at Yale School of Public Health and was subsequently hired as an assistant professor there, studying a variety of vector systems and beginning her work in tick-borne diseases. She now runs an eco-epidemiology lab at Columbia that focuses on integrating a wide range of empirical and analytical tools to disentangle the ecological and environmental drivers for the emergence of vector-borne diseases, particularly tick-borne. Current research focuses on the role of pathogen interactions, host community composition, climate and land use changes, and the epidemics of multiple tick-borne pathogens in the U.S. Thank you very much. Thanks so much for having me. I'm very excited. I heard all of you do very exciting things. So I'll be talking about ticks. I know there's two people at least that are interested in ticks. And in particular in urban areas and in New York City, but we're probably going to expand to other areas. So I'd like to start by talking about Lyme disease epidemiology a little bit. Lyme disease, for all those that don't know, it's transmitted by Ixodes scapularis. Borrelia burgdorferi is the bacterium, and it causes a variety of symptoms, from a rash to more severe manifestations like arthritis, carditis, and it can become quite severe and debilitating. So it is the most highest incidence vector-borne disease in the U.S., and the incidence up to 2020. The number of cases are up to 40,000, but it's estimated there's about 10 times more. The CDC estimated 400,000. Sorry, estimated that it's actually 10 times more than that. So it is really an important disease. So the Lyme disease has been expanding geographically. So this is a map from an estimated distribution in 2000, and that's 2017. So it has really filled up the Northeast region and the Midwest. And it's also expanding south, basically in all directions. And it's also expanding into urban areas. These are cases in New York City. Most of these cases are due to travel, but about 50% on Staten Island, which is over here, part of New York City, are actually not travel related. So these people are getting infected, are getting tick-borne diseases on Staten Island. So that's a map my student Meredith Van Acker did when she started her PhD, just to get a feel for what's the situation with ticks in the city. So the crosses are no ticks present, circles are very low numbers, so those may have been dropped by birds, and then the higher prevalence and density of ticks are on Staten Island. So the rest of the work we mostly focus there, but I'll tell you about another project, an expansion to that. There's also, as you know here, other ticks such as Lone Star ticks, which are very common over here and transmit other set of pathogens, and the dog tick and the longhorn tick. That is also expanding in distribution in multiple directions. So far we have not identified any human pathogen, although it is a vector of zoonotic pathogens in Asia, and it also has been found to be potentially transmitting a cattle pathogen. So the transmission of Lyme disease the cycle is composed of a wildlife dimension and a human dimension. So of course, hosts have a fundamental role in terms of their availability that is determined that is associated with their composition as well as their movement. So if the right composition of hosts are present and they interact with the ticks and the pathogens, we measure what we call the hazard as the density of infected ticks, so the nymphs in particular. So those are going to be the stage that are more likely to bite humans and transmit Borrelia burgdorferi or other pathogens. Of course, abiotic conditions need to be appropriate as well for them to survive. And so humans may become exposed through the ticks and develop Lyme disease. And that's linked to human behavior, outdoor activity patterns, or their protective behaviors. So this system also has feedbacks in it. So humans can, for example, feed wildlife, and I'll talk about that more later. And these resources can modify the host community. So that can then feed back into the system. And so I'm calling this the wildland-urban interface, which I'll describe in a minute. But it's basically this interface between humans and wildlife that can result in transmission of tick-borne diseases. And that's all, of course, happens in a landscape. That's actually Staten Island looking into. So a framework, so I'm going to describe the framework that we use in my lab to study vector-borne zoonosis. We look at how climate and land use affect the distribution of the ticks, the hosts, and the transmission of the pathogen. So the interaction between mice and the pathogen and the ticks determines R0. And this interaction is a very critical one, and it varies a lot depending on the composition of the host community and even the activity time, the time of activity of the ticks. So if all the interactions are correct, then we're going to have persistence of Borrelli-Burgdorferi, that's R0, and that's what determines what we're calling the hazard, which is a potential source of harm. So all of these are eco-evolutionary processes that are independent of humans. So that's why all of this makes up the hazard. And then we think of this hazard as almost like an environmental hazard towards humans that then become exposed through their behavior and ecology and behavior, and then depending on the vulnerability, they may develop disease. So this framework of hazard, exposure, and vulnerability, again, it is possible because this is a zoonotic disease. So humans are not part of the transmission cycle. So it only goes basically in that direction. Humans need to enter areas where there's hazard to become infected, in contrast with many other diseases, of course. And so in this framework, we look at the hazard, the risk is, we define zoonotic, the risk is the product, I mean, not necessarily linear, but of the hazard, the exposure, and the vulnerability. So those three things are what determines the risk. And then we can divide that by your capacity to mitigate your risk by taking, for example, taking any precaution like using caricides in your yard or repellent, things like that. So climate and land use really influence all of these processes at the same time. So they can affect the ecological cycle, the hazard, the exposure by changing people's behavior, increasing, for example, with heat and with co-vulnerabilities as well. And so the complexity of the cycle is something I wanted to emphasize, you know, when we're trying to ascertain whether it's climate or land use that is driving, for example, increasing in cases, the process can be happening at multiple levels, which makes it really hard to predict what really the outcome is going to be of climate change or land use change. And of course, these things also interact with each other and feed back into each other. So given this background, the questions I want to talk about today are what's the role of, in particular, I decided to focus on one aspect because there's so many components, but I'll talk about habitat fragmentation in particular and how it affects those multiple components. Then I'll talk about some frameworks with which we can integrate the components. And then we're going to look at them, how they vary across a gradient, for example, an urbanization gradient. So then the idea is to get one factor and start to understand how all of the other elements vary among each other and over space. So a little bit of history of the invasion, because that's important, of course, to understand the disease. There's a claim that deforestation, that reforestation, bringing back the forest, caused the Lyme disease epidemic. Basically in the 70s, most of the cases were coastal, and 100 years ago, there were very limited numbers of deer. So if you look at a time scale, this is an approximate time scale for Connecticut, but it applies to a lot of the Northeast. There was reforestation followed by a decrease in deer numbers, and then there's an increase in Lyme disease. Of course, these are a lot of years from each other. I'm not saying those are causal, but we do know that deer are a keystone host for the cycle. So deer are necessary because they host the tick, and the tick reproduces in the deer and the adult ticks require a blood meal from deer to maintain the cycle. So you do always need deer for Lyme disease to exist, pretty much, with maybe one exception that I'm aware of. This is giving a little more detail of where the levels of deer populations in pre-European settlement times, and hunting basically brought them down to extremely low levels. In the 1900s, deer would show up in the paper, like somebody saw a deer was like big news. And then they rebounded very quickly after hunting was regulated. And now they reached what we perceive as very high numbers, but if I look at that, they are kind of pre-European settlement numbers. So maybe they're not, I mean, what is natural numbers, but they've been kind of rebounded, and in many areas they are higher than at least what's considered ecologically stable. So one idea then is, okay, reforestation is quote unquote bad, it brought back the deer, it brought back Lyme disease. And there's an opposing view that they're not mutually exclusive, is that it is the fragmentation of forests what's causing the epidemic. And what does fragmentation do to the system? It does two things. So one, it affects the zoonotic hazard, because fragmentation in general will change the host community. And this is classic fragmentation theory, that species that are living, interior species that live in core habitat will become extinct after you fragment the landscape. And then there will be some species that are more adapted to edges that will benefit from it. So for example, mice and deer are quite adapted to edge habitat, so they would be benefiting. And in smaller patches, there will be predator and competition release. So the mice that are highly competent for Borrelia, for ticks and for Borrelia, would benefit from not having competition and predation and increase in very large numbers. So that's how the theory goes. That's been called the dilution effect, the idea that biodiversity is protective, and then when biodiversity declines, you start getting more of these species that tend to have more pathogens. And the reasoning behind it is whether this is a general principle. The reason would be because some of these species have very high reproductive rates, so they have, because of the demography, they have a lot of offspring, so they can transmit the pathogens, and they also they invest less in immune defenses. So small rodents are typically one of the species that are considered highly common. However, I'll be talking in a minute about one of the things that is not described here. Is like what's happening in the middle here. So these are, island biogeography type of ideas of fragmentation, that these are islands that become fragmented, but there is certainly a matrix, and we'll be talking about what's in between those islands. So this is pretty complex, a cascading effect. And then there's another effect that is in a way a little simpler, just the fact that people are living in those places increases the exposure at this wildland urban interface. It's just that the fact that you're living close to forests where the deer and the ticks are, you're more likely to encounter them. So these two things happen at the same time. The same process can generate those two effects, but they're different, right? And if you were to decide what to do in terms of control, are you going to try to modify the ecology of the system, or are you going to come to terms with, if you live in the forest, you're going to get ticks, and that's exposure, and there's nothing can be done. So that's another, or you have to protect yourself individually or spray or things like that. So in terms of the operational or the significance of that, it's important to separate the two. And then some people with their ticks. So this wildland urban interface is actually a metric that has been described for fire. It's used for fire. And it's developed to understand areas that are high risk for fire, and that are actually described by having intermixed. There's two types. There's one where there's intermix, where you have humans living in proximity to nature, and there's also humans living next to nature. So there's two types that are. So that's well defined, and it's been used a lot to prevent fire or to predict fire. So if you look at the map of the wildland urban interface, this is a percent of the population that live in the wildland urban interface. It matches the Lyme disease map pretty well. I mean, not exactly, but it does match, and it does make sense ecologically, right? That we have this fragmentation. But it's interesting how they have a number that you can map. And a dream of mine some days, like have a map like this, but with these exactly ecological conditions that predict Lyme disease at the level of the landscape. That's kind of where we're going. Because this is not perfect fit, but try to get it to be something that defines this is a Lyme disease type of really high suitable habitat. So we talked a little bit then about how does fragmentation affect the two components of risk. And so we want to integrate them somehow. We want to understand how they relate to each other. So I mentioned that they are different. Actually, in some cases, they're opposing trends completely. So this is a classic paper that described this fragmentation causing a more hazard. They call it the density of infected nymphs. So when the patches, this is an supposedly urban area, but not as urban as some of the areas I'll describe now, but then the smaller the fragment, the more ticks you have. So there's like a negative exponential decline in the hazard with the area. And this other paper by Brownstein, this is Brian, and this Allen, and by Brownstein found a similar effect. But then when they look at Lyme disease cases, they found the opposite effect. So it was positively associated with the sizes of the patches in the landscape. So that means there's something that is not going, we really need to understand what's driving these two things, because they can't really go even in opposite directions. So like some years ago, we worked on this paper. We looked at an area. This is an endemic area in Westchester County where it's highly endemic for Lyme disease. And this is work by Max McClure in my lab. And he worked on this index where you can look at for each of these units. So one of the issues that we have is that when you look at a tract, let's say a census tract, and you have a certain number of Lyme disease cases, the process that occurred, and you have population for that tract, but then a lot of the ecological studies on patch dynamics are at the level of the patch. So it's a very different unit. So you need to really, if you want to understand what's happening in the tract, you want to see how, patches, basically these two things are related. So this index, what it does, it's a sum for each of these units over an index of exposure. In this case, we're saying exposure is proportional to maybe the edge, how much edge there is between the patches within your tract. Or you can say it's proportional to how much green areas there is altogether, or it could be the surface. So there's different metrics, and we tried a number of different ones. But this means that these edges are the interface, right, where people will encounter ticks. And then the hazard. In this case, the hazard is a negative exponential, just going with the description of, deriving from that paper, that's a negative effect with fragmentation. Again, as I explained before, small patches will have more mice, more ticks, more pathogens. And so we looked at different metrics, and we did find actually that this hazard was the most, so the models that have hazard explained described in this way were the most predictive for Lyme disease, and that was pretty consistent. In terms of the exposure, there were different metrics that were also consistent, so we couldn't find one that was better. But the interesting thing is, in this case, we have like a combination of both of them that was predicted for Lyme disease. So that was with real data. So that was real Lyme disease cases that were predicted by the landscape. Then what you can do with that is then create different landscapes and start asking questions. So you can simulate landscapes that have different, in this case, forest cover or fragmentation. And this we use an index of percolation. So it's like a way to artificially create landscapes that are more clumped or less clumped. And then we can put a grid on top of it, and then we can imagine that each of these is our units now, like it would be like a census tract, but these are all artificial units. And so with this, you can start playing around and say, well, how is forest cover important, or how fragmentation are important? These two things are both important, and they both vary at the same time. It's another factor that's different to separate. So doing that, we found that actually 20% occupancy gave you the most Lyme disease. So if you have a county where 20% was occupied, is according to that model, that's most Lyme disease. But the fragmentation also matters. So if you had more fragmentation is the largest number, that's the probability of having a tick. So the more fragmented it is, the more risk you get. And then the less fragmentation ameliorates that. So then we can start understanding some of the processes. At this point, it's just patterns. So we're just seeing what relationships are there. So the next step will be to see what might be driving that, what's causing this to be 20% and those numbers. But we have a starting model. So to start thinking about what might cause those relationships, we actually work with this couple natural human systems. We got a grant from NSF that's called that. It's called IDAS now. And it developed a more dynamic model of what the system might, to explain mechanistically what can be driving those processes. So we are looking at an area where there's this is our zoonotic risk in here, and it's from part. So there's parts, there's a transmission cycling parts that they can spill over or into human areas as well. They might or not come back like that, but it's mostly from the parks and people live next to them that they can acquire ticks. And then the hazard becomes depending on people encounter ticks and depending on their activities or the protection that protects them against ticks. And then this is a really interesting part because we're trying to see what people do. What are the investment decisions based on their risk? Do they invest in personal protection? Do they spray their house? Or do they tackle or they reduce deer or try to kill the ticks on the deer? So depending, and these will operate at all different scales. So this one will operate at the whole area at a more general scale. This is just you, this is like your family. And so by thinking about that, the feedbacks then into the system would be different and would operate at different scales, depending on what intervention you're using. So then the number of ticks, so of course this relates to the community and just the funding sources. And so what can happen here is that some of these feedbacks, we can see an example of a feedback would be if you do an intervention, you spray in your house, let's say, then there's less ticks spilling over into your house. They stay here, but there's less in your house. So there may be less hazard going on to you. So you decide then, okay, I don't need to take protection, I'm spraying, I'm good. And then, but then maybe the spray is not working. And so then you still have IDAS and so forth. So this is what we call like these feedback loops that the system might be caught in. And we cannot identify them by just looking at models. So these kind of models, agent-based models, allow you to identify those feedbacks. And this is an example of what an agent-based model would look like. So we would have agents that have rules of behavior and movement, will be humans and wildlife, and we have a landscape. In this case, we have the zoonotic cycle happening in this particular patches, people living, so we can choose either realistic landscapes from the field or simulated landscapes like we talked about before. So deer move, humans move, and then we see what happens. And so here so what I'm going to talk to you about now is a little bit of which metrics we collect from the field to inform this model. So what you want to do is get parameters from the literature or from the field and inform the rules of movement of these individuals, and then also have data to calibrate the model to make it as realistic as you can. So we measured those three things for the hazard: the landscape composition and the connectivity, how the species assemble, and the spillover into backyards. And then in terms of the risk, we looked at human movement and risk perception. I'll tell you all about those, or at least some part of those things. So then once we have that information, so we understand how this works, we can run them, as I said, across those landscapes. Those are by postdoc Jonathan and Pilar, who was by postdoc, now is a professor, and run them across your fragmentation gradient. So ultimately then what we want to see is how these things vary over that gradient. And the hazard, for example, goes up probably in areas that are more trees, but there may be a dilution effect. They may go down at high diversity, potentially. Then exposure may increase here, but may not have the same shape. This is a tick now. And vulnerability may be higher, maybe in more urban areas. Maybe people have other vulnerability, socioeconomic, or of other diseases or of other kinds. So the shapes of this curve is what we're gonna simulate using the model. So we talked a little bit about integration. So one, there's the statistical integration by looking at functions that describe each of these by observed patterns, putting them together, and then more mechanistically understanding the processes underlying those, for example, with agent-based models. So now let's see how we can do this in an urbanization gradient. So why do we care about urban areas? In urban areas, the ticks will be transmitted in parks. And so, of course, parks and green spaces provide a lot of ecosystem services. As you all know, they moderate the climate, reduce pollution, help mental health, and many advantages. But there might be a disservice. When deer become New Yorkers, they may bring ticks. That's a kind of an unfortunate event. But because we love deer, but So how does the composition and configuration of these urban green spaces may affect the hazard exposure and vulnerability for tick-borne diseases? So we published a paper where we tried to synthesize a little bit of what could be happening in that gradient. So one of the things is that, of course, that we talked quite a bit about this landscape transition, and we use a little different terminology than the patch matrix, the patch and patch isolation. Because what happens is first you have, a very green area and you get into this wildland urban interface where initially you have these perforated areas, so a house there in the middle of the forest. And it's not, so is it a patch or you don't know which one is the patch? Is the forest a patch or the house there is the patch? And then you keep moving into more and more urbanized area, and then the patches are clearly the green spaces in a very urbanized area. So the processes or the mechanisms that are affecting the system are different. So here we have like an ecotone, we'll call it intermediate disturbance, where it actually could be beneficial to the hosts because they have a lot of resources and humans are exposed a lot. And so the composition of the host community may be critical here, the numbers of species of different hosts. Now, when you keep moving here, what patch size and isolation become dominant because now you have a barrier. You have the city that is a barrier for the animals. So movement, being able to reach those patches becomes dominant, which it may not be the case here. Animals may be able to navigate, this area pretty well. Here they hit a threshold where they have to cross the freeways. They have to get to those patches. So looking at those processes in terms of the landscape, and of course, host movement is highly determinant of that. So hosts change the sizes of their home range, their behavior becomes more tortuous in cities, they use more human resources. A lot of things happen in the host communities in cities. And then humans, of course, have different types of exposure. So here it would be recreational, in parks, in natural areas. Here will be mainly domestic around your home. And then here there will just be no risk. And in this case will be more recreational in these parks or due to travel. So all of those things are changing at the same time. So if you were to decide, well, so people have asked me, like, how would you design green spaces to have a lot of green space, but not have deer and not have ticks, for example? That's something that we could want to do. And there's a lot of literature on this, like it's been called land sparing for a design of natural reserves. You have a chunk of green area. What should you do? Just have one big area that will accommodate a lot of interior species, or you have many, many smaller areas. That's been the SLOS debate if you have done ecology. So this is a debate for design of urban areas, right? You have a central park there, or you have like a lot of pocket parks in other areas. So the dilution effect would predict that that there will be more zoonotic hazard here, right? There's small patches that have a lot of mice and ticks, and they're good for that. And this one will have less zoonotic hazard and also less exposure. The idea, humans typically move around 500 meters. They're lazy, they don't walk any more than that. And that is statistically proved. And I remember not wanting to walk more than 500 meters. So if you live here and it's more than 500 meters, you don't go to the park, basically. And so people that study human behavior have shown that. And so the humans are lazy, they don't go, animals may not get there, so there might be less hazard and less exposure. Now, do we really know how animals and humans move? Are there exceptions? So that's kind of where the question comes in, is we really need to understand how animals are using this habitat and how humans are truly using it. And they may drive to the park, even if they're lazy. So the other question, a lot of this, in terms of the landscape, a lot of the studies have done this idea of patches that are isolated, and then they have more roots where not in agricultural areas. All of the references, at least I've seen for Lyme disease, are in agricultural areas. Now the matrix here is fundamentally different to an urban matrix. So you have, this matrix varies a lot depending on the crop. It may go through times where there's nothing there and so forth. Of course, urban areas have a lot more barriers to animals and a lot more resources. So there's, raccoons can live on trash, just sit by the trash can and the rats, and that's all they need. And so these areas may be very suitable if there's a nice trash can there for raccoon. So that's what I feel that it hasn't been really done. It has not for ticks at least, but a true understanding of how does the urban matrix modify the traits that are needed to survive there. So in terms of, let's go a little bit more about the host community. We talked about that, it's going to change, but let's think a little bit about the life cycle of the ticks. So the ticks have 4 stages: egg, larva, nymphs, and adults. And the larvae come out in August, typically, and feed on anything that's there. The ticks don't, I mean, some of them do, but not a lot of preferences, especially they mature. So they might feed on amplifying hosts, so small rodents that are very competent, and that's been shown in the lab. You know, these animals tend to produce a lot of infected ticks, or what they've been called dilution hosts, or hosts that are not as good as transmitting the pathogens, like raccoons, squirrels, and opossums. So if they bite a lot of these, they are going to get infected. If there's a lot of these animals in the environment, then there's a dilution effect. So the tick will bite anything, so proportionally, if you have more of these hosts, that dilutes the infection overall. So the larvae drop off, and then the nymphs emerge months later, and they will encounter same host community. If they're not infected, they're going to infect the next one. Then they drop off, and then they emerge as adults. The adults feed on deer, as I said, exclusively, pretty much. And I forgot, in this figure, there should be deer here too. So deer are dilution hosts. Their blood kills the bacteria. So if an adult feeds on them, that's great. Great for the ticks. There will be a lot of ticks. But if a larva or an immature feeds on a deer, they don't get infected. They're dilution hosts. So they decrease the infection, increase the ticks. They have this double effect. Okay, so given that, the way the dilution effect theory has been set up is that the fragment, so we have a very diverse community in a pristine environment, supposedly, and then there's communities disassemble. So humans start chopping off the landscape, it gets fragmented, and some species don't survive. And eventually you end up with a mouse or a highly competent host-dominated community. And as I said, the assumption that the one remaining is highly competent comes from this idea that it's debatable and debated whether competence is associated with those traits related to demography or other traits. There's lots of paper now about traits and infection for all sorts of things. But the small, the demographic seems to be one thing that is common, although there's a lot of biases and discussions about that. Now, for this system, we forget there's deer. So deer also need to be present, and they're probably present throughout in different abundances. So what does a city do to the community? First, it will establish a connectivity threshold. Deer cannot get to Central Park. Thus, we have no ticks in line, luckily, in Central Park. So that is a limitation that was not in this original idea. Yeah, there's a lot of mice, but if there's no deer, no mice. The other thing is that these hosts also adapt to humans. And if you look at these guys, they're not very much in pristine environments, as much as all over here. And so they adapt to humans, and they really dominate in human adaptive environments. And if you have doubts about that, I'll show you some pictures. This is the whole community as one in somebody's yard. Everybody, all the dilution hosts. The raccoons having fun, the cat eating mice and eating cat food. Everybody in New York City, I don't know about here, eats cat food. Every animal. Everybody. So deer and all of the animals eat all the cat food that's provided to cats. And sometimes they fight for it. Very much. Not necessarily the cat is happy about that. And this is cameras we have like studying urban wildlife. So that raccoon is studying. Those are comes in Central Park. So not from pristine environments. Very human adapted hosts. And these are the dilution hosts. So that's something to think about. I mean, whether these guys live in tick habitat or not is something to determine, right? This opossum was very upset. I don't know why. Somewhere else. Yeah. So a lot of so this is a situation in highly urbanized areas where I guess maybe the community is actually more diverse or different from what you would predict, and it hasn't really been fully studied. So when you look at exposures, then I mean we talk about the host community that may become more mouse dominated, but maybe not. Maybe we have all this host eating trash that also contribute to the tick cycle in highly urbanized areas. Then in terms of exposure, we know probably in Manhattan, everything will be travel related because there's no ticks there. Then we go through areas where people visit parks and they're green enough. This is another transect we're actually studying. These are ticks, the circles are ticks, a number of ticks. So we actually went through, well, this is Staten Island, and then we're expanding this over. No ticks here, but already you start seeing ticks in Queens in New York. So the deer got some, well, these might be dropped by birds, but these are definitely brought by deer. And so people visit parks and their activities may expose them to ticks. And then, of course, in areas, more green areas where people live in that kind of house, that will be more peri-domestic. So then the exposure also switches over space. And so our study on Staten Island, after Meredith found all those ticks there, I was happy because I came from Yale and there were no ticks in the city. I also had Columbia, so I was happy there were ticks. It's not very nice of me, but it's like I need to work on something. So Staten Island became the center of my world, and all the New Yorkers are not very happy because they don't like Staten Island. They're like, well, it's a really tick. So these are the kind of forests of Staten Island. It's pretty amazing. You know, they're just like very pristine forests. And they are very proud of their forests. They're really beautiful green belt of vegetation, and then they get all those ticks there. So the design of how we study it, we wanted to look at the critical things that have been proposed in terms of landscape and ticks. So one, we wanted parks that vary in connectivity. So we selected those parks that were different size and different connectivity. And I'll show you in a minute, my student did a map of connectivity. And then we defined what we call this ecological neighborhoods. So we looked at houses in 200 to 500 around the park. So those are the ones that are likely to get ticks from the animals going into their backyard. And then the houses vary. So there was a connectivity at the scale of Staten Island and the connectivity of each of the houses to the park, and we call that permeability. And then the participants were recruited. So we surveyed people around there. So this is the design. So parks that vary in connectivity with each other at a large scale and impermeability at a small scale. And this is just a little bit of all the stuff that we did and photos of all the people that did all that work. So these are tick host abundance and infection. We did trap animals, trap goat ticks, and also camera trap. Colors on the deer. The deer there have colors, so we have animal movement. We can understand how they use the landscape. That was also worked by Meredith. Then these are just two postdocs that did a lot of the coordination. Then we looked at the backyards, students doing that in the houses. And then we looked at similar things as this, but in the yard. So we collected ticks, we put cameras, and we have these tubes that capture hair. So you see the animals, and we ID the animals by the hair. So at least we know presence absence, because putting this is very hard in someone's house. So then we had the tick app, which I'll tell you a little bit more about it later. It's an app where we follow people, like if they allow location services, and then we also ask them questions and they send us pictures of ticks. And then we went to the parks and we looked at people and we sampled ticks, and we tried to see to what extent people are using habitats with ticks versus other habitats. So at that scale, see what the exposure would be. So going back then to this map, so I told you how all the data was collected. So let's see some of the results. The composition configuration is the first one. So looking at the parks that had had ticks, or look at the correlation between ticks density in the parks and covariance. We found that parks that are surrounded by trees are more likely to have ticks, but those that are surrounded by water or soil are less likely. So that means they're more isolated. And then most importantly, we found that this metric of connectivity, which is called flow centrality. So it's similar to using an electric circuit that you can circulate through the area, looking at the resistance of the different landscapes. So if you remove this park, this is the most connected one. If I remove the whole electric circuit, basically, so it's kind of like the way to measure that. So this connectivity was related to nymph infection prevalence and the density of ticks. So this is opposite in a way, right, to the idea that the most isolated parks are the ones where you're going to have more mice, more ticks, more infection. The other part is a lot of data of data movement, which I'm not going to have time to show you, but these are home ranges of all the deer that were monitored. And so we have rules of movement for the deer. So we know the proportion of the deer selection, the criteria they use to select certain habitats versus others. So a lot of information that then we can put in those HMAs models. And then sampling hosts with camera traps and also small mammals. Meredith found that in very connected parks have more mice and deer, and less connected parks had more squirrels and raccoons. So again, proving that less connected is where those hosts are. These were related to more ticks collected, those with less. And deer was associated with another parasite transmitted by the black-legged tick, Anaplasma phaeostetophilum, and mice with Babesia microti, so these two are hosts of these other parasites. Actually, for Borrelia burgdorferi, we found no significant association because it is more commonly distributed, so we didn't find a significant association. But this tells us that there is a link, a cascading effect of fragmentation on the host, the ticks, and we had also different species of ticks, but I'm not showing those results. So that means, again, fragmentation affecting the host community, and then the tick and the pathogen communities. So the third part, so I told you the first one was the host composition, and then the cascading effect into the landscape and the connectivity. And now is this, we're going to look at the spillover. So if you are near a park that has ticks, how likely you are to get ticks in your yard. So what are the characteristics of the yard? So there's attractants, like the cat food that everybody eats, repellents and evaluating conditions that determines whether the hosts come to the yard. So we sampled all of these different features that we thought might influence the probability of hosts coming and the suitability of the backyard. And we did this pair design where we had, these are all mouse traps in the forest, and then we had neighboring houses along that edge. So we could see how the animals navigate that interface at that small scale. So we found actually this is that the richness of species was actually higher mostly in yards than in parks. I don't like the richness very much as a metric because just a number of species doesn't tell us too much, but it was kind of in the opposite direction that you would expect. In natural areas, there should be more. The dilution effect idea would propose that would be more richness in those areas. Connectivity was positively associated with abundance of deer, foxes, and rabbits, and negatively with groundhogs and skunks. So actually, groundhogs and skunks were more common when the house was less connected to the natural area. And red fox and eastern gray squirrel were related to their presence in the matched park. So we looked at so that means that they were more dependent. So only some areas had foxes, and those are the foxes that came to the houses. Versus some of these other animals were present very broadly, so they're not necessarily linked to their presence in the park next to them. Then we looked at the proportion of all the photos that we found of these animals in the parks and in the yards, or the proportion of tubes with hair of those animals. So it's not the same animals, but it's just a proportional, like community-wide comparison. So we found that some of the animals are equally likely to use both the shrews, raccoons, and skunks, so they kind of go back and forth. Small animals are more common in parks, and some animals like the possum and the groundhog, like I said before, and the cats were more common in the backyard. So those are animals that live in the backyard. So in terms of moving the ticks, so we're interested in animals that really will navigate that interface, and we're able to, such as raccoons, that can bring the ticks over to your backyard for your residential exposure. So that's why we did this, and then so we also have data from radio tracking that we've not ready yet, from GPS tracking of these animals, that we will also be looking at kind of the same thing, but for the same individual. These are just community-wide metrics. So this means that this really this supports this idea that this intermediate disturbance, the idea of the intermediate disturbance hypothesis. And there are several papers now that have found that there is more diversity where people live in their backyards, because you're providing resources. The animals have both, the best of both worlds. They have the forest, they have open areas, so there's more light, there's more vegetation, and they have all of humans' resources year round. So that's something that continues to be other people find. And so in terms of the presence of ticks, because that's ultimately what we're interested in, black-legged ticks was highly predicted by the connectivity of the park, how could the presence of deer and logs, brush logs, like having wood there, and negatively associated with a fence, luckily. So something was actually stopping this guys to some extent. But so similar with Lone Star ticks is same kind of thing, although Lone Star ticks were associated with their presence in the park, not so much by the host. So parks that had Lone Stars are more likely to spill into people's homes. And then the longhorn tick was only associated with those log piles, magic log piles that I'm like, just clean them up. So they're probably related to a metric of something else, of habitat, because they're just small, maybe, or I think they're correlated to something else, the type of yard, or something else that people that have log piles or other things go on there that we still don't know. But anyway, this one, not associated is very adaptable. So it's in all different habitats, and fences don't stop it, in part because it moves in raccoons, that raccoons can move adult. The raccoons have adults of these, but not adults of black-legged tick. So it's still deer, the main one for black-legged tick. So we also captured animals and tested that. Okay, so we have landscape affecting the community of hosts. Yeah, the connectivity being a factor, especially for deer, the assemblages being modified depending on the resources in the city, and the spilling over into yards depending on aspects of the park, but also of the yard itself. So for the last part, I just want to talk a little bit about risk, just a little of how human movement and behaviors are affected. We have tons of data on that. I'll just touch on a couple of things. We do parks, as I mentioned, park surveys that take up, and also surveys in people's homes. For the park surveys was actually kind of fun, just watching people what they did. And then the probability of tick encounter. So we estimated a probability of tick encounter and looked at the correlation between the time spent in all the different habitats. So that can give you, if we were to follow a person, which we don't, we just watch a person being there for 2 hours. With a tick up, our next step will be to try to follow the person and have a cumulative amount, cumulative exposure, basically, as you would have, with people. That's been done for mosquitoes. Gonzalo started that, but it's never been done for ticks, like just to estimate what's your exposure. So some things we learned by the same people, not the same people, but in the same parks. We asked them questions, and then most responded, but the parks is where they might get ticks, but they believe that they didn't have risk. So a lot of the times they're like, yes, there's ticks, but I don't have risk. The cap didn't change across the parks. So basically some had a lot of risk and some less, but people were unaware or responded the same things. And so basically they didn't tick check. So most people were just really not aware of that there were ticks in the park they were in. And so the tick up lets us look at the behaviors. We asked people about their activities and preventive behaviors. And this is what the tick up looks like. You can download it for free. That is really cool. And we have a hard time people doing it because you do have to answer a bunch of questions. So a lot of people just don't go back past this. But once they do, and we're going to pay them. We're starting to pay them to do this because they just don't use it. So we asked them to survey every day for 7 days at least, even if they didn't find a tick. So we got a log of positives and negatives, which is what we need. And then it's hard to make people understand. We need the negatives. It's important. So mostly they report a tick, but which has been, So a lot of things we've learned. So it's a collaboration with the Midwest Center. We know that Midwesterns are more outdoorsy, things like that. And they tend to use more prevention activities in general. So these are kind of broad patterns. The interesting thing during the pandemic, we saw when we asked people what are your activities, are indoor, outdoor, recreational, or very domestic activities. And so we found that outdoor activities went up. This is where the lockdown, and we had data for the same date a year before. So we kind of could compare. We found that the peridomestic activity went down hugely, and the activity in parks went down. So there was this exact switch, and then it's controlled for seasonality. And so there's been studies that show, or papers that show that there's more people visited parks more, but at least our population spent a lot of time in backyard, which is also something that happened. And we found 30% more tick reports than the previous year. So people were being more active outside and getting more ticks. But then, of course, Lyme disease wasn't being reported, like so many diseases that fell through. But we have an indication of that. So that was kind of a little project during 2020. It was like, well, outside, we had the tick. So the tick encounter reports is another study that we're working on, and so we can monitor people's movements, so we can see where they got, how much time they spent. So these are the people that actually submitted that they found a tick. And so what proportion of them have moved out of county or out of zip code? So that's something that you always wonder where they found the tick. So we're not going to know exactly, but we know there's probability that they may have found it outside. And so we asked them to report where they went, and 12% told the wrong place, or 70% told the wrong zip code. I mean, they maybe didn't know. I don't know if there's motivation to lie there, but so these kinds of checks we're checking how the information the tick app gives us. Then people that say they do recreational activities spend a lot of time in grasses. So we have that GIS data that monitors that. And doing paradomestic activity, they were more like cultivated habitats. So we know what type of habitats they spend time in. And these are tiny numbers, but this for me is the most interesting thing. So those that found scapularis had spent tons of time in forests. Those that found amblyoma had spent time in grasses, and dog ticks, they spend time in cultivated lands, and that matches the habitats that are more common for these different ticks. And they're very small numbers, but we're hoping to increase that. And that gives me also confidence that that kind of makes sense. So summary for the tick app, and we're wrapping up. The exposures are context specific. So this was very broad comparison across regions, and then also activities across years. And we need to have high resolution data. So we need to start looking more carefully to highly urban and county. Actually, we look at urbanization and have different patterns of travel, the users. And those that have unknown encounters, in general, so they didn't know where they encountered them, and we knew they had spent time in forest. So that's probably where they got it. And so we can have all this information. And so last one is the household surveys. That we have, again, millions of questions, but these are one where just estimating whether people were repelled, what they do in terms of protecting themselves against Lyme disease. And I'll show you, because I want to show you a little bit of a simulation. One ABM, so we've been starting to run the ABMs, the agent-based models. This is for behavior, for adaptive behavior. And so in this one, we try to measure the percent of people that encounter a tick per day, and we can simulate adaptive behavior. So if you encounter a tick, you change your behavior. And we try to look at potential different behaviors that people could do and the outcome. So this is just an example of the model with the park, the yards, so people become exposed to ticks, and the model specifications. And so by running this model in different landscapes, the landscapes that I showed you before, we saw that in general, so this is fragmentation and this is forest cover. And so if there's low forest cover, more fragmented actually increases human-to contacts. But in areas with high forest cover, the relationship is non-linear and it's more complex. So we can start building this exposure map based on some. In this case, there's no specific behaviors, but these are derived from the simulation. But we're going to start feeding some of the real behaviors to the model. So those are the kinds of things that we can come out with from the model. And the other thing is when we model adaptive behaviors, we can see the percent of tick encounters, how they changed the encounters. So for example, if people did nothing, the encounters are still very high. And people avoided the park, so we give those rules of movement. The agent, every time it finds a tick, then it doesn't go to the park anymore. And so that's the most effective. And then, there's the others are intermediate. This is kind of an obvious result, but we can quantify that because we could have the actual hazard in the environment. So we have a number, the probability of encountering a tick, that's a hazard. So we can build that into the model, and then we can have realistic, ideally, people movement with a tick up or other data to simulate that. Other things that we're going to add is some social vulnerability index, of course, and then we are looking into data on park visitation to start building some of the models. So the next project we're starting is expanding from Staten Island to this gradient I showed you before. So now we want to go back to this landscape. So we were focused on this ecological neighborhoods around the parks, but we're going to go back to thinking about these green spaces. With all the data now that we have on the tools, we have the visitation, we have the app and all that. And so in this gradient, we expect are not and you win because it's a network of camera traps. So we have camera traps distributed. It's a nationwide network of people that put camera traps in urban gradients. And so the idea is that R naught will stop here, we'll find the threshold for deer, and then we are going to start building the ecological neighborhood and link them up to have finally landscapes that are basically at different levels of risk. So this is kind of a vision of the system running through quote unquote thresholds. They may not be linear, but you can think of landscape connectivity being connected enough basically to have deer. If there's enough deer, there's an R naught from the ticks more than one. If there's enough ticks, there's R naught from the pathogen is more than one. Once a certain threshold is surpassed, people start encountering ticks. Then Lyme disease cases start being reported. People start perceiving risk. That happens usually later. And ideally, people that may trigger individual protection, and then even more will trigger area-wide interventions. So these are some of the wildlife aspects, these are the human aspects, but I can envision this happening across this spatially, but also temporally, and in terms of management. So finding these thresholds is kind of what we're after, really understanding if it's what's the shape of those relationships, and if there's some point where you switch to a different equilibrium. So in summary, and highly fragmented landscapes, connectivity really enhances rather than reduce the abundance of deer and mice. And so that has a cascading effect. Human use of subsidies really shape the abundance of these dilution hosts. They increase actually dilution hosts. And that reverses this idea that preserving those hosts, we should conserve biodiversity to help, because these are not hosts that are part of a biodiverse environment. They're like living with us. So it's kind of a confusing concept. So the spillover that increases in the yard with the connectivity with forested landscapes, and we can think of filters, ecological filters of yard features and other factors that filter a subset of the community that can actually occupy those backyards. And human movements are nonlinear and complex, as we all know, and we still have a lot to do with human behavior, as you can imagine. And so these are all people like once it's updated in my lab, the TICAP team, the CNH, that's a team, and I think. Any questions?
Oh, great, thanks so much. You mentioned briefly birds, and I'm just wondering how much of a role you think there is for birds in picture dispersal.
Yeah, so they do have a role for sure. So we have other studies with birds. Yeah, we did a lot of things with birds. So birds that during the breeding season, they can be part of the zoonotic cycle. So, and then we have a study of Block Island where the ticks fed, we estimated that larvae fed a third of their own deer, a third on mice, a third on birds. And so the birds also carry different genotypes. So we're looking at that on Borrelia burgdorferi. So they seem to be adapted to some extent to them. So that has an influence. And then there's a long distance dispersal, of course. So in the city, actually, they could have more of a role in bridging, right, across the parks. Yeah, I over-focused on mammals, but we're hoping to add birds as another layer. I mean, then there's a very long migration dispersal. And that, I think it only is relevant when you have a lot of birds, like to Canada, to certain sites, because the birds migrate not at the best time for migration of the timing of the phenology of the ticks. And also they don't carry too many ticks and not infected with Borrelia, some species more than others, but so in terms of migration, it's more limited. We think it's mostly, when you look at the patterns of migration, are more like diffusion, like terrestrial, although for Borrelia, there are some jumps. So we think for Babesia, there's no jumps. So I didn't show that one, but so we think it's more mice related. But then with Borrelia, if you look at the correlation between autocorrelation across the sites, you see that sometimes there's, like indicates there's more long distance dispersal is consistent with Borrelia being in birds, but Babesia is very rare in birds. So that's the other thing we do. Sometimes we compare the ticks or the pathogens, because then you get some, more realistic things. But yeah, I should have mentioned more about it, but yeah, they definitely. Yeah. Yes.
Is there some kind of competition between different tick species, and does that affect the risk of transmission in any way? Like you mentioned, there are parks that have multiple species of ticks.
We don't know if that's a big question and we're interested in that, but so the deer have all the ticks and all the stages all at the same in the summer when you store it in certain times. Is if there is, it would be on the deer probably. We can't think of anywhere else. So it might be some immune mediated, if there's some immune component that affects the other tick more than it affects you, I guess, because sometimes, that would, like the most of the hosts, or many of the hosts have some sort of immune response to ticks, even mice, but it's very low. It only happens when they have a lot of ticks. But if there's some cross immunity, anti-tick might have an effect. But so far, we think it's mostly habitat driven. So far, so for example, in amblyomma is more tolerant to dry conditions. It's in pine forests in Long Island and New Jersey. Is in pine barrens. You see more amblyomma. You see very few. So we think it's more climate and habitat, but it's hard to prove that, right? Unless you get like a lot of deer and you put them artificially, it's just very difficult to explain. Yeah.
Do you know why that deer aren't able to actually have like the Lyme disease bacteria infect them?
So yeah, they have it. So the complement systems, so it's an innate immunity of the deer. It kills the parasite, and it's not uncommon, especially so in Europe. There's two Borrelias, the Borrelia cellia and Garenia. One is mouse adapted, one is bird adapted, and their complement kills basically the other one. So we know that it occurs in animals, and then also birds vary in their competence. We have a study of that, and it was a role of the complement, but also some antibody response as well. Yeah, it's I don't know why would it happen in deer and not other? Well, evolutionarily, I don't know. Yeah. Yes.
Thank you so much.